The yeast-2 hybrid system is a molecular genetic approach which makes use of the necessity of protein-protein interactions in virtually all biological systems. This method is used to identify genes whose protein products interact with a protein of interest. During the test, two vectors are produced that encode hybrid proteins. In the first one, the cDNA coding for the protein of interest called the bait is bound to the DNA binding domain, which is responsible for binding to the upstream activation sequence UAS. This is called the bait hybrid. In the second vector, cDNA coding for a protein that can bind the bait is fused to an activation domain, thus creating a prey vector. There are multiple copies of the second vector with a cDNA library cloned to each of them. This way they all encode for an activation domain fused to a different prey domain. The prey domain is the domain that binds to the bait domain. Then the bait and prey vectors are transfected into engineered yeast cells. If both hybrids are expressed in the transformed cells, the bait and prey domains of the proteins can now interact. Successful interactions result in forming a functional transcriptional activator that is necessary for the transcription of the reporter gene. If no interaction occurs between the bait and prey, the DNA binding domain and activation domain of the transcription factor will not be activated, hence resulting in no expression of the reporter gene. Note that because one end of the entire protein complex binds to the upstream activating sequence, while the other end facilitates transcription initiation of the promoter, the distance between the DNA binding and activation domains must be quite flexible for this to work. Lastly, specific gene expression markers can be used to select out for cells that express both vectors and where the two proteins interact to allow for a specific gene to be expressed.
A paper titled Recruitment of the Autophagic Machinery to Endosomes During Infection is Mediated by Ubiquitin was recently published in the Journal of Cell Biology. This paper aimed to investigate the importance of ubiquitin in the process of autophagic sequestration of invading bacteria. The yeast-2 hybrid method was used twice in this experiment. The yeast-2 hybrid assays were performed using the Matchmaker GAL4 yeast-2 hybrid 3 system as previously reported. A strain of Saccharomyces cerevisiae was co-transformed with the cloning vectors containing the inserts of interest. The figure that can be seen depicts the results found in one of the yeast-2 hybrid mapping experiments showing the ATG16L1 FIP200 interacting domains. ATG16L1 is an autophagy-related protein and FIP200 is required for autophagosome formation. Figure A and B depict the mapping of the binding sites between the two proteins, and C and D depict the results from an additional yeast-2 hybrid mapping experiment showing which ATG residue the FIP200 fragment binds to.
Some controls need to be taken to ensure the minimization of false positives and also negatives in the results when using the yeast-2 hybrid method. False positives can be minimized by using negative controls, such as water, to substitute plasmid in some of the transfection. This helps to ensure that any yeast growth is due to a success of both plasmid transfection and also protein interactions. The incorporation of antibiotic resistance gene into the plasmid and the utilization of growth media containing antibiotic also acts as a selective marker that would prevent growth of any non-transformed cells. Also, false negatives can then be minimized by using positive controls, such as transfecting yeast cells using plasmid that only contains bait and prey proteins that are known to interact. The expression of genes should also be under the control of inducible promoters, such as IPTG inducible promoters. This helps to prevent any failure of expression of genes that are due to either a lack of control of the promoter or due to a faulty plasmid construct.
Despite the controls that have been made, yeast-2 hybrid methods still involves other possibility of false positive and false negative identification, hence limiting their usage. Other false positives could result from the overexpression of bait or prey protein. Also, some proteins might specifically interact when they are co-expressed. However, in real physiological conditions, they have never been present in the same cell at the same time. Another source of false positive is the interaction between the prey protein and the reporter protein, or with the membrane anchors fed to the bait. Finally, proteins that are not correctly folded can also show some non-specific interaction. The site of expression of yeast-2 hybrid vectors takes place in the nucleus. If the test proteins are not localized in the nucleus, two interacting proteins may be found to be non-interacting. Furthermore, the interaction between the two proteins Yeast-to hybrid method is often not symmetric. The feedback of the hybrid may cause direct hindrance that impedes interaction. The lack of post-translational modification in the yeast system can cause false negative results. Very transient interaction may also escape detection.